Hello friends, this is Bright's Microbiology and today we are going to see about DNA and its structure. So DNA is commonly known as deoxyribonucleic acid. So the acronym is DNA and it is a polymer of deoxyribose nucleotide. So this deoxyribonucleotide is a polymer which is composed of many monomeric units, namely the DAMP, DGMP, DCMP and DTMP. So we all know DNA is made up of 4 nucleotides, namely adenine, guanine, thymine and cytosine, out of which adenine and guanine are purines and thymine and cytosines are pyrimidines. So similarly, the adenine base pairs with thymine and guanine base pairs with cytosine. That is, the purines and the pyrimidines base pair with each other. No purines and purines can base pair each other. Similarly, no pyrimidines and pyrimidines forms a base pair. So this rule was given by Chargaff's rule of DNA composition. So he observed that all the species that he studied in the late 1940s showed that the residues had equal number of adenine and thymine, similarly equal number of guanine and cytosines. The A and T had double bonds of hydrogen, the G and C had triple bond of hydrogen, and this is known as Chargaff's rule of molecular equivalence between the purines and the pyrimidines in the DNA structure. This was not immediately realized as a significant rule, but when Watson and Crick proposed the theory of double helical structure, this played a major role there. So it was considered to be a strength for Chargaff's rule. Now let's dive into the double helical structure of the DNA. The double helical structure of the DNA was proposed by James Watson and Francis Crick in the 1953, which I earlier mentioned. The elucidation of the DNA structure is considered as a milestone in the era of modern microbiology. The structure of the double helical structure is similar to a structure of the ladder which is twisted. So let's look here. This shows a structure of the ladder which is coiled around like a double helix structure. When this is unwinded, you can clearly see a structure which resembles a ladder. So this is also known as twisted ladder structure. So let's look into the salient features of the DNA. The DNA is a right-handed double helix structure. So this consists of a two deoxyribonucleotide chains, which are also known as strands, that is twisted around each other with a common axis. You can see a blue strand and a brown strand running around the axis with the nucleotides placed. So this is a deoxyribose nucleotide chain and it runs around each other in a common axis. The two strands are anti-parallel in nature. So one strand runs from 5' to 3' and the other strand runs from 3' to 5' . So this is compared to a parallel road carrying traffic in the opposite directions. So the width of the double helical structure is 20 angstrom, which is 2 nanometer. Each turn of the helix is 34 angstrom, that is, it is 3.4 nanometer with 10 nucleotide base pairs within it. So each nucleotide is placed at a distance of 3.4 angstrom and each strand has a hydrophilic deoxyribose phosphate backbone. The sugar phosphate backbone is hydrophilic. Hydro means water and philic means loving. So the sugar phosphate backbone is water loving, but in the case of the nucleotides that are stacked inside the nucleophosphate backbone, they are hydrophobic, that is, they don't attract to water. So this is the hydrophobic and hydrophilic nature of the DNA. So the two polynucleotide chains are not identical, but they are complementary to each other due to the base pairing. This we already saw in the Chargaff's rule, that A and T binds together and G and C bind together. So they cannot be identical. That is, if the one strand contains A, the other strand must always contain T. So this is the rule. No strand can contain A or G or C or T. That is not possible. The two strands that are held together by hydrogen formed by complementary base pairs. The AT pair has 2 hydrogen bonds while the GC pair has 3 hydrogen bonds. So we have adenine and thymine which is bound together by 2 hydrogen bonds, and the guanine and cytosine pair which is bound together by 3 hydrogen bonds. So automatically the G and C is stronger by 50% than A to T. The hydrogen bonds are formed between a purine and a pyrimidine only. No purine then purine can bind together, or a pyrimidine can do so. So the spatial arrangements can be A to T, T to A, G, C or CG. So the complementary base pairing in the DNA helix proves the Chargaff's rule. The genetic information resides in one of the two strands known as the template strand or the sense strand. So the double helix structure has a major groove and the minor groove. The major groove is wide in nature and the minor groove is narrow in nature. The proteins interact with the DNA at these grooves without disrupting the base pairs or the double helical structure. So these are the salient features of DNA which was proposed by Watson and Crick. Conformational changes of double helix. The variation of the conformation of the nucleotides can be associated with the conformational variant of the DNA. So the double helical structure of the DNA exists in 6 different forms, that is A to E and one more that is known as Z. So among these, B, A and Z are the most important forms. So the B form is the double helix structure which was described by Watson and Crick that we discussed above. It is the most dominant form in the physiological conditions. The B form, or as we already discussed, it has 10 base pairs spanning at the distance of 3.4 nm with a width of 2 nm. The A form is also a right-handed helix such as the B form, but it contains 11 base pairs at turn, but there is a slight tilting at the base pair by 20 degrees away from the central axis. So the Z form is a left-handed helix and contains 12 base pair per turn. The polynucleotide strands of the DNA molecules moves in a zigzag fashion and hence it is known as Z-DNA. It is believed that the transition between the different helical forms of the DNA plays a significant role in regulating the gene expression.
Other than these, there are 3 different forms of DNA. They are the bent DNA, triple stranded DNA and the 4 stranded DNA.